All right, guys, let's finish this last lecture before our exam. So this is finishing the slide packet from Tuesday, really mainly covering 9.1 and some of 9.2 and 9.3. I only expect you guys to know 9.1 for the exam. You don't have to study 9.2 or 9.3. You are welcome for that. But I am going to cover some of the GMO stuff just because it's interesting and I think you guys would enjoy it. All right, so just to refresh you, we went over this now a few times. This slide is explaining the central dogma, basically the idea that genetic information flows from DNA to RNA to protein. And there are two steps. Ugh, I'm sorry. That's terrible manners. I would pause and re record that, but I think you guys will appreciate the fact that there's a yawn in the middle of the lecture. It's real life. But there are two steps that are involved in this flow of genetic information. The first one is transcription, where you're transcribing the information in DNA into a molecule of mRNA. And then there's translation, where you're translating this nucleic acid language of A, G's, C's, and U's into an amino acid sequence. So you're translating from nucleic acid to protein. All right, so we already covered transcription. Look through the slides, through your textbook, watch some of the videos provided with the lectures. If you are wanting to go over that, we're going to pick up with translation, talk about mutations, and then spend some time talking about GMOs. All right, so translation translates the mRNA into protein. I know we kind of went over some of this in the lecture, but I'm just going to start fresh. I think it'll be better for us. And I switched the order of some slides because I think it'll help you out. So you go from RNA to protein. And this is part of the meal analogy that we've been looking at. In that analogy, a protein is going to be the meal that you are making with the recipe. The ribosomes are going to be the chefs. They're the ones who are actually making the protein. The amino acids are the ingredients. That's what the protein is made of. And the tRNA are the tools that help bring the amino acids to the ribosome. So how does this happen? How does the information on mRNA actually used to build a protein? So here we've gone from DNA to mRNA using the first step of the central dogma, transcription. And we noted in class that the mRNA is simply built using complementarity. The fact that A's in RNA, A binds to the T, G binds to C, U binds to A, C binds to G, and A binds to T. Right? So using that complementarity, RNA polymerase is going to make the mRNA strand. And then the genetic code is actually translated using codons, which is a 3-nucleotide sequence that encodes one amino acid. So when the mRNA is actually being translated into protein, the mRNA message is read off 3 letters at a time, a codon at a time, and those 3 letters specify one amino acid. So here AAG in the mRNA actually specifies the amino acid lysine. So let's look at how these codons specify an amino acid. So again, a codon is a stretch of 3 nucleotides that code for one amino acid. And basically, each amino acid is paired with a tRNA that can recognize specific codons. So each tRNA pairs with codons via an anticodon. So here in the figure, you have your yellow tRNA, which is an RNA molecule, a transfer RNA molecule. And it has a sequence called an anticodon that is complementary to the codon. And so the tRNA will recognize specific codons, but the tRNA is actually bound to a specific amino acid, depending on the anticodon sequence. So that is how tRNAs actually translate RNA to amino acid, is they basically couple a specific codon to a specific amino acid. And then you have this little table here, which is really just, you don't have to learn it, or really even, I don't expect you to necessarily know how to use it, but the idea here is that you have different codon sequences. So on the left, you have the first base of the 3 base codon. So on the left, you have the first base. It can either be a U, a C, an A, or a G. On the top, you have the second base, which can be a U, C, A, G. And on the right, you have the third base. So basically here you're making all the different combinations of 3 letters that you can make. And you'll notice that there are amino acids that match to different codons. You'll notice that actually there is more than one codon for each amino acid. A lot of them have 2 codons. Like if you begin at the top left of the chart, you have UUU and UUC that code for phenylalanine. If you go to the right, to the next column, you have serine, and there's actually 4 different codons that code for that. So there's a little bit of overlap, but the point is that if you go to the top left again, the codon UUU will always specify the amino acid phenylalanine. I can't even say that. Phenylalanine. If you go to the next column, at the top row, you have UCU. That codon will always specify for the amino acid serine, and so on and so forth. So each codon is specific for an amino acid. And now you know the way that a codon specifies an amino acid is that it actually can bind to the anticodon of a tRNA that is matched to a specific amino acid. So the take-home message of this slide is that tRNAs are special RNA molecules, transfer RNA molecules, that are bound to specific amino acids and recognize specific codons using their anticodons. So they are the molecule that actually bridge the gap between mRNA and amino acids. They are truly the translator. And these are used by ribosomes. So we went over this a little bit. We'll go over it again. Ribosomes are large multi-protein and RNA hybrid enzymes in charge of protein synthesis. They're composed of 2 subunits, a small subunit on the bottom and a large subunit on the top. The small subunit attaches to the mRNA and positions the mRNA properly to begin translating it. And the large subunit will then chomp down like a hamburger bun, right? You have the small subunit is a small part of the bun and the large subunit is a large part. And this large subunit facilitates tRNAs binding to codons and then binds amino acids together to form a polypeptide chain, which is just a chain of amino acids. And here is an actual 3D reconstruction of a ribosome, where you can see the RNA in gray and purple up top, gray and blue, sorry, and the protein is in purple. And you can see in the small subunit, the RNA is in green and the protein is in purple. Okay, so how do tRNAs and ribosomes work together to build a protein? This is kind of where I think I lost 90% of you, so hopefully I'm going to regain those of you that got lost. So the first thing is that the amino acids and the tRNAs that are floating in the cytoplasm are bound together in a very intentional way. So tRNAs that recognize specific codons are bound to particular amino acids, to their appropriate amino acids. And once they're bound, the tRNA will dock onto the ribosome if there is a complementary RNA codon present. So basically, if the codon on the RNA, say whatever 3 letters are up next, if those 3 letters and the codon are complementary to the anticodon of the tRNA, to the sequence on that tRNA, the one that's coming in is UCC. So if there's complementarity there, that tRNA is going to dock and position itself right next to the growing chain of amino acids. And then the ribosome is going to join the amino acids together to form a polypeptide, which again is an amino acid chain. And then after that one tRNA is loaded on and the amino acid is added to the chain of the polypeptide chain, the ribosome moves on to the next codon, the next 3-letter sequence, in order to receive the next tRNA. And it does this until the ribosome reaches a stop codon, which is a sequence that tells the ribosome that this is the end of the line. And then no tRNA can base pair there, and the newly synthesized protein is released. And as this newly synthesized protein is released, this protein, this chain of amino acids, is going to fold and take its final job. Take its final shape, which will allow it to perform its job or function. And then the subunits of the ribosome separate and are ready to reassemble and begin translation of another mRNA. So there's a cool little video on YouTube that I'd love for you guys to watch. I'm a big fan of videos because they help take these two-dimensional images that are static in your textbook and on these slides, and they animate everything for you so you can see how these things happen in as we think they happen in real life. So it just helps you get a better idea of what's going on. So watch that video. You can download the slides and click on the link on the slide. Now let's look at where the central dogma occurs in the cell. So where transcription and translation occur in the cell. So in eukaryotes, the transcription occurs in the nucleus. Because remember, the DNA, you never want to move it out of the nucleus. The DNA needs to, that's why you have a nucleus, is to protect the DNA. So if you're going to make a copy of the DNA, you're going to be making it in the nucleus, in the central office. And once you make a copy of the DNA in the form of mRNA, now you can take that mRNA out of the nucleus, so that mRNA actually is going to be exported out of the nucleus into the cytoplasm, where translation is going to happen. So ribosomes do not exist in the nucleus, they exist in the cytoplasm. So the RNA has to be exported out of the nucleus into the cytoplasm, and then ribosomes can actually translate that mRNA message. And in prokaryotes, they don't have a nucleus, so actually transcription and translation occur in the same compartment. So they do it all, and they actually, you can see in the figure here, that they do it simultaneously. So as RNA polymerase is transcribing the gene onto mRNA, a ribosome is already loaded onto that mRNA and is translating that mRNA into an amino acid sequence. Okay, one last thing for chapter 9.1. How do mutations work, and what are mutations? Mutations are changes introduced to the genetic code, and it's like changing part of a word in a recipe. Ultimately, mutations alter the function of a protein. Oftentimes, a mutation will destroy the function of a protein. And this is because the change of DNA sequence results in a change of amino acid sequence. Because now you know that DNA, the genetic information in a gene, gets transcribed onto mRNA, and that mRNA has codons, 3-letter chunks. So if you change one DNA nucleotide, that is going to possibly alter a codon, change the codon to where now you've changed whatever amino acid was specified by that codon, and that can lead to malfunction of a protein. So here in this figure, you have the DNA sequence of a gene, and the gray box there is highlighting a codon, CTA. Now, when mRNA polymerase comes along, it's going to transcribe that CTA codon onto an mRNA as GAU. You'll notice that GAU is complementary to CTA. And then that GAU codon specifies the amino acid asparagine by using a tRNA. Hopefully that makes sense now. Now, suppose that letter T that is paired up with an A, you introduce a mutation and you change that T to a C, which is going to now be paired up with a G. So you're basically changing the base pair from TA to CG. But because you have changed a nucleotide in the codon, instead of being GAU, now you have GGU, you have changed the amino acid specified by that codon to glycine. And when you change an amino acid, that can actually affect the way that affect the shape of the protein. So here you can see that this mutated protein has a different shape than the normal protein. And in this case, it just so happens that this change in shape makes a protein non-functional. It can't do what it's supposed to do. And mutations can happen in a few ways. Randomly, when DNA is being copied, just like when a scribe would introduce an error into a text, when DNA polymerase, this is not the polymerase that's involved in transcription, that's RNA polymerase. DNA polymerase is involved in replication, so think mitosis, think cell division, the interphase. When the cell needs to replicate its DNA, it does it with DNA polymerase. So DNA polymerase is also like a scribe because it makes a copy, and sometimes scribes make mistakes. Sometimes when DNA polymerase is replicating DNA, it'll make a mistake and introduce an error into the text or into the sequence. So mutations happen randomly. So live long enough, even if you live in a bubble and you touch no carcinogen, no harmful chemical, you will still get cancer because mutations happen randomly, because DNA polymerase makes mistakes. And mutations can also happen when DNA comes in contact with harmful chemicals, such as smoke, UV rays, and other carcinogens. So essentially, what these cancer-causing chemicals and particles are doing is they're introducing a mutation into the DNA sequence, which can eventually lead to a different amino acid replacing an amino acid, which can lead to changing the shape of the protein to where it can't work. Now, mutations have different effects. You can have a missense mutation, where essentially you're going to change a nucleotide or a base, and that's going to replace the amino acid. So that's the example that we just saw on the previous slide. Change one letter of the DNA sequence, and that change in one letter affects the codon which codes for that amino acid, for an amino acid. So you replace an amino acid. And that leads to a non-functional protein most of the time. You also have a nonsense mutation, which I'm not showing here, but basically you also change one letter of the DNA sequence, and that's going to create a stop codon that tells the ribosome to stop. So sometimes you can get a nonsense mutation early in the gene, and so the ribosome will only translate that first part of the gene, of the mRNA. Once it gets to the nonsense mutation, which has created a stop codon, that ribosome is going to stop translating and get off the tracks, right? It's going to dissociate. So these nonsense mutations produce short versions of proteins, which is not good because you are missing a huge chunk of the protein. You also have silent mutations, which I'm not showing here either, which are a change in a base, in a letter of the DNA sequence, but not in the amino acid. So actually, even though you're changing the codon, you're changing it to another codon that specifies the same amino acid. Because if you go back to that table a few slides ago, you'll notice that some amino acids have 4 to 6 different codons that code for that same amino acid. And then lastly, you have frame shift mutations, which are really bad because they introduce or delete a base, a letter in the genetic code, and that either creates a stop codon or just shifts the whole frame of the genetic sequence, of the gene sequence. And that's not good, right? That'll throw off everything. It can create stop codons, or it can just totally change the amino acid sequence. All right, and I don't expect you guys to know the differences between these, but just know that these are different ways in which the change of one letter of the DNA sequence can result in a change at the amino acid level, at the protein level. All right, so a little bit about GMOs, genetically modified plants and animals. A transgenic organism is formed by the incorporation of a gene from one organism into the genome of another species. These are what are called genetically modified organisms. So, for example, in this picture of a fluorescent cat, this is a real thing. If you were to have a black light party with this cat would be glowing green as you guys are partying. And the reason this cat glows green is because what the scientists did that created this really amusing creature was they took a green fluorescent protein gene from a jellyfish and put it into the cat genome to where now the cat actually expresses this gene in all of its hair cells. So the whole fur of this animal is fluorescent green. Sounds pretty cruel, but actually the cats are fine. They don't notice a difference. Uh, they just glow in the dark when you put UV light on them. Now, obviously, the main way we encounter GMOs are not genetically modified pets, although I will say that will become more and more common. But the main way we encounter GMOs are through food. So 85% of US corn and soy is genetically modified. And because of a lot of processed foods use corn syrup and soy and vegetable oils, a lot of these processed foods are actually going to contain genetically modified stuff. Because again, 85% of corn, or we get corn syrup from, is genetically modified. Soy as well. So a lot of the processed foods contain GMO products. If you go to Whole Foods, though, you'll see that a lot of their foods have stickers, labels that say non-GMO food. And it's a commitment that they have made to sell food that does not have genetically modified, that is not using genetically modified ingredients, which is good. But the reality is that genetically modified foods really aren't any worse for you than foods that haven't been tampered with. I'll be making a plug a little later about an interesting documentary called GMO OMG. It's on Netflix. You can watch it, it's free. Um, and they specifically look at genetically modified soy. And they do mention so, one of the main downsides of eating genetically modified soy is that this soy is resistant to weed killer. So farmers can use a ton of weed killer to keep the weeds out of their fields. So those foods might contain a little bit more chemicals than organic food, non-GMO soybeans. But the amount of chemicals on that genetically modified food is still below the limit set by the FDA. It has to be because it has to be approved by the FDA. So in the end, you're not getting more than what is allowed on your food. It's still a very minimal amount that most likely won't affect you in any significant way. So really, the whole GMO thing is blown out of proportion. I think the documentary actually is a little bit anti-GMO, but it presents a pretty cool argument and good evidence for and against GMOs. Really cool. All right, so I want to walk through with you guys one of the earliest and most classic examples of scientists messing with genes from organisms in order to help the food industry, in order to increase production of the food industry. So in the early 1980s, scientists were successfully produced recombinant bovine growth hormone, or RBGH. Now, normal BGH is a hormone in cows that increases their body size and their milk production. So basically, it's like a steroid for cows, right? And they normally naturally make this. So what scientists did was they took the cow gene for BGH, inserted it into bacteria, and then used that bacteria to make recombinant BGH that can then be harvested and injected into cows in order to increase their milk supply. And they found that RBGH increased milk volume by about 20%. So that's a huge profit that's efficient for for the milk industry. And about 1/3 of cows in the US are injected with RBGH, right? So now more and more milk companies that care about not using GMO products are going to label their milk with a label saying these cows were not treated with RBGH or RBST. It's the same thing. Um, Again, expressing their commitment to not use genetically modified organisms, which is great. I appreciate the idea of being as natural as possible. And the FDA, what the FDA has done is they have looked at cows that you have been treated with RBGH and cows that were not treated with that, and they found no detectable difference in the molecular composition of the milk from these two different cows. So it's not like if you drink milk from cows that are treated with this RBGH protein that you're going to get cancer, right? Again, GMOs, not that big of a deal. In this case, the cow itself is not GMO, but the RBGH protein that's being injected into the cow is genetically engineered. But yeah, that's what they do it. So how did they do this? How did they make recombinant BGH? So 3 simple steps. First step, remove the gene from the cow chromosome. So you take the cow DNA and you use molecular scissors that can cut. These molecular scissors are called restriction enzymes. They can cut the gene from the genome and they leave sticky ends. And then you take this cut gene that has sticky ends and you insert it into a bacterial plasmid. And a plasmid is just a small circular piece of bacterial DNA that is separate from the bacterial chromosome. So it's just a little piece of DNA the bacteria carries. So what they do is they cut the plasmid with the same restriction enzymes, creating the same sticky ends, and then paste the cut BGH gene from the cow into that plasmid. So now the bacteria has this gene, this recombinant BGH gene, and can actually express this gene and make the protein, recombinant BGH protein. And what we mean by that is just genetically engineered. It's a gene that has been removed from its original genome and combined with another. So then you insert the plasmid into bacteria, and now you have bacteria that express this RBGH gene and make loads of RBGH protein that can be harvested and injected into cows. So these bacteria essentially are now RBGH factories. So the only reason you're taking the gene from the cow and putting them in the bacteria is because bacteria divide quickly and they can make a lot of this protein. So essentially, this bacteria is going to be making loads and loads and loads of this protein that now can be injected into cows to help their milk production. That's really all RBGH treated cows are for. It's just the reason they inject this protein is so that they can make more milk. Crazy thing that has been coming out recently is something called farming, which is production of GMOs that produce drugs to treat or prevent human disease. So it blends pharmaceutical and farming, hence farming with PH, right? So scientists have engineered tobacco plants which produce human proteins found in blood that help with blood clotting, goats like the one below that are producing human blood clotting proteins in their milk in order to help with people that are anemic and can't properly clot their blood. Their concerns, obviously, and it's really ethical concerns of using animals. Basically, these animals are being bred in order to create pharmaceuticals for human diseases. So, is it ethical to do that? And you're actually modifying their DNA. So, with the goat, for example, you're putting proteins, you're putting genes into the goat's genome for human proteins, and these human proteins are being made in the milk and then harvested, right? So, is that ethical to tamper with animals' DNAs in that way? So, if you're interested in learning more about GMOs, I really recommend this documentary that I mentioned called GMO OMG on Netflix. Very interesting documentary on the potential hazards of GMO soybeans and other foods. Worth checking out, really interesting stuff. All right, last thing we'll end with, super brief, cloning humans. What the heck? Is this true? Is this real? Do people do this? Are scientists crazy and already cloning humans? Most likely not. Those are probably just conspiracy theories. But what has happened is in 2013, US scientists performed nuclear transfer with human cells. Nuclear transfer is just replacing the nucleus of an egg from a woman with the nucleus of an individual being cloned. So here in the figure, you can see that the first step is to remove the nucleus from a body cell of the person to be cloned, and then transfer the nucleus of that cell into an egg that has had its nucleus removed. And essentially, what you've done is you've created a zygote, which is that first step after a sperm and an egg fertilize. Except here, both sets of chromosomes are coming from the individual that you're cloning. Instead of having chromosomes from mom and chromosomes from dad, the sperm and the egg combining, here you're inserting a nucleus into the egg from a person that has already two sets of chromosomes. So you're essentially cloning that person. So in 2013, scientists did this and produced an embryo with 200 cells. So they didn't let it go past the 200 cell stage. And they used these cells for therapeutic cloning, which is basically using stem cells to create healthy tissues or organs for transplants. So the idea here is that, say, the person that is being used for cloning here has some genetic disease and needs maybe needs new blood cells. And so what they can do is they can create an embryo that has a lot of stem cells, harvest those stem cells that are going to be the same genetic material as the person that has the disease, because you've done this nuclear transfer thing. You're going to take these cells, and now these cells can actually, you can use them to make more blood for this person, to treat this person with their own healthy blood, basically. So pretty crazy stuff. Obviously, eventually, if you let the embryo grow, the embryo could actually be implanted into a woman, the uterus of a woman, and that embryo could grow into a baby, and you could technically clone a human. Now, has that happened? No, that hasn't happened. Um, Honestly, don't think it's going to be happening anytime soon, mainly because there are laws in place that prevent this from happening. Um, so yeah, but who knows? Maybe in the future. And honestly, I would agree with most of you guys listening to this that this would not be a good idea. I don't think anyone truly thinks it would ever be a good idea to clone a human being. So hopefully that's far from now, and hopefully it'll never happen. All right, so that's it for our online lecture. Again, you're only responsible for 9.1. The rest of it is just fun stuff that hopefully has taught you a little bit about what GMOs are. So, online test reviews posted. Put any questions that you may have on the review, about the review. Post that on the discussion board, the discussion thread on our Blackboard page. I'll make a little online lecture on Thursday or Friday and post that for everyone to review with. All right, so hope you enjoyed the lecture. Have an awesome, awesome day, awesome week. Bye.